Good afternoon. Good afternoon. Happy Wednesday. Okay, still waiting for folks to come in. Look at the website, the Canvas unit for genetics. We're going to finish up that video that we saw. We're going to talk about Mendelian genetics. So if you would log into Canvas. Pull mine up in just a second. Actually, I'm going to try to queue up that video so that we start off where we left off. Okay, okay. The sun catcher from Custom Cages is the perfect outdoor enclosure to enjoy the element. Creation should be. Okay. I can't do both. So let's see how am I going to do this. I was trying to queue up the video and then show you where you needed to be in Canvas as well. To do that, I've got to pull up two different pages. So let me do that. All right, because I indicated that we started the video and we left off in the middle of it. But I'm going to go a little further back. And what is that noise? Okay. Still waiting for some folks to come in. Missing like about 5 people. And Keone, you are in, but it looks like you're popping in and out. Like you got some internet issues going on there. You're in and I keep admitting you. You're on the list, but you're not on the list. All right, so I'm going to give it a few more minutes for folks to come in. But
Genetics Primer, Mendel and
oops.
Modern Genetics Terms.
All right, well, I can't stop it once it starts. Oh, okay. If I don't press the button, it won't go. All right, so what I want you to do is to look at this while we're waiting on a few more people to come in. Take a look at this one, and then we will go back to looking at the other one. And I'm going to step away for a minute and I'll be right back, but everybody should be looking at this.
Gregor Mendel is known as the father of modern genetics. Mendel was an Austrian monk, and he lived from 1822 to 1884. His genius was that he noticed differences in peas being grown in the monastery's garden and applied the scientific method to determine the cause of these differences. Mendel chose the garden pea Pisum sativum because peas were easy to grow, they had a short generation time, they had a number of contrasting traits that were easy to distinguish, such as short versus tall, or white versus purple flowers. And they also normally self-pollinated but could be cross-pollinated. Mendel was able to control mating this way by removing the male or the female flower parts and cross-pollinating with a paintbrush. Normally peas do cross-fertilize or fertilize themselves, but he didn't want this to happen. Mendel didn't really know about inheritance. No one had great ideas about it at that point in time. We knew what cells were, but we did not know about organelles, protein synthesis, or even genes or chromosomes yet. All Mendel had to go by was physical traits and his knowledge of flowers and statistics.
Okay. All right, let this last person, these numbers keep jumping back and forth. I don't know what is happening with Zoom. People are in and people are out. All right, but hopefully all of you who are here will be staying here. All right, and those of you who just signed on, please be on camera. Thank you so much. Got one more coming. One more coming. All right, so I'm going to switch over here because this one's already queued up. So we're talking about genetics. Let me see if that'll switch on your screen. Yay. All right, we had started this one and I went back a little bit, but I'm not going to go all the way back. But again, we're talking about chromosomes, and the different sections on the chromosome are called genes, and the genes are going to code for different traits. So I'm going to start it from here and
which is
continue.
quite complex and determined by interactions of many genes together. In fact, the ability to taste PTC or not may involve some other gene interactions. There's even different ranges for how bitter the chemical may taste because there may be more kinds of alleles than we'll mention. More about that later. But since we do know that the ability to taste PTC or not taste PTC is at least heavily impacted by a specific gene, it does make it powerful for genetic classes. One thing I have found so interesting is that my parents can both taste PTC. So why can't I? Recall that humans have 46 chromosomes. Chromosomes are made up of DNA and protein. It's a condensed unit of DNA. My whole genetic code is represented by these chromosomes. You inherit 23 chromosomes from your mother and 23 chromosomes from your father. Here's all 46 of them right here. As you can see, there are 23 chromosome pairs. Each pair has one chromosome from one parent and one chromosome from my other parent. If we focus on one of these pairs of chromosomes where the PTC taste sensitivity gene may be found, we can see an area where the PTC taste sensitivity gene could be. Let's assume this is the locus, where the PTC taste sensitivity gene is found. See how it is pointing to a specific area here? That's because it's on an area of the chromosomes that refer to a specific gene that codes for a trait. Now, remember how this chromosome is from mom. This one is from dad. Each parent contributes an allele, which is a variant of a gene. An allele is a variety of a gene, a form of a gene. The alleles could be the same form of the gene or different forms of the gene, but regardless, in this case, they're forms of the gene involved with PTC taste sensitivity. So, if PTC taste sensitivity is being used as a one-gene trait example, and as we mentioned, it may not be that simple, then your DNA code has a gene related to PTC taste sensitivity. Together, the two alleles you inherit, the forms of that gene, determine the trait of tasting PTC or the trait of not tasting PTC. That gene is involved with coding for taste receptors on your tongue, and the receptors you have can make a difference for whether you taste PTC or not. The alleles are typically represented by letters. Since this is all about tasting, let's use the letter T. But wait, it matters whether I represent it as a capital or lowercase letter. If I use a capital letter to represent an allele, it means that it's a dominant allele. If one or both of the alleles you inherited for a trait are dominant, then it will be expressed. More about that later. If I use a lowercase letter to represent an allele, that means it's a recessive allele. Recessive alleles are typically not expressed unless there is no dominant allele present. Now remember that you have two allele copies, so the combination you could have here could be big T, little t, or little t, little t. These are called genotypes, your genetic makeup. Genotypes can help determine a phenotype, which is a physical characteristic. You'll notice when writing genotypes, I put the capital letters first, if it contains a capital letter. That's not because the order matters, it's a formatting formality that capitals are written first. It turns out that being able to taste PTC is a dominant trait. That means the phenotype, which is a PTC taster, is due to a genotype that includes at least one dominant allele. So which genotypes can taste PTC then? Well, big T can. Both of those alleles are dominant. So can big T, little t, because remember, it only takes the presence of one dominant allele. In fact, the only genotype in this simplified example to not be able to taste PTC would be little t. So obviously that is what I am. I am the little t genotype, which results in my non-taster phenotype. But my parents can taste PTC. So what genotypes would they have to be? Well, if they were both big T, that wouldn't be possible. If one was big T and one was big T, that still wouldn't be possible. Remember, you have to get an allele, a form of a gene, from each parent. If my parents do taste PTC and I do not, then my parents have the genotype big T, and their phenotype is PTC taster. Punnett squares can be used to determine the probabilities of offspring having certain genotypes, which then can be used to determine their phenotypes. But Punnett squares are for another Amoeba Sisters video. Before we end, one more thing to mention. In this example, the dominant trait of being able to taste PTC is more common than the recessive trait of not being able to taste PTC. And one could jump to an assumption that dominant traits are more common, especially since it only takes the presence of one dominant allele to show up in the phenotype, at least in Mendelian inheritance. But the dominant trait is not always more common in a population, because it's possible that the dominant allele itself is more rare. That can be the case with some forms of polydactyly, that is, being born with extra fingers. Some forms of polydactyly can be a dominant trait caused by the presence of at least one dominant allele. However, the dominant allele may not be as common in the population, and the condition of having extra fingers is generally rare. Well, that's it for the Amoeba Sisters, and we remind you to...
Okay, so this is where we're going. We're going to be talking about traits and chromosomes and dominance and recessiveness, and how to draw a Punnett square the way they just did. And I'm going to do this one.
At Grand Canyon University,
Then they frown
we
upon
believe in
showing
equal
back two
opportunity,
videos
and
back
the
to back.
American
That was a
dream
leftover
starts
from the
with
last
purpose.
class.
Professor Dave here. Let's
All
talk
right,
about...
Professor, hold on. All right, so we're going to talk about Mendel and what we call Mendelian genetics. Why they call it Mendelian genetics is because Gregor Mendel is considered to be the father of genetics. And so his way of looking at all of the laws of heredity are what we study. Now, we have since come up with some more modern things, tweaked his theory a little bit, but for the most part, whatever Mendel's laws were, those are the laws that we're going to follow. And hold on one second. I'm going to come back to, since I'm switching things at the same time here. If we go back to our vocabulary list, oops, no, I could have stayed right there. What we're going to look at, actually, I'm going to go back to that page. All right, so we're looking at Mendel's laws, and down here to the bottom. So we're going to look at it. So I just told you who Mendel is. He is the father of genetics, and he came up with all kinds of rules and a lot of vocabulary. So we're going to look at these three principles down here, the principle of dominance and recessiveness, which is what she was just talking about. Dominance means that the characteristic that you have is stronger than a recessive characteristic. So while they're both coding for the same type of thing in your body, like eye color, one will show up. It's like standing in front of somebody. You have two genes and one stands behind the other one, and you can only see the one that's up front. So the one that's up front is what we would call dominance. Okay. All right. All right, for our late folks, we are in Canvas and we are looking at genetics. All right, the second one is the law of segregation, and that's going to talk about the way our chromosomes behave during mitosis and meiosis, and then the principle of independent assortment, where they are distributed, the ways they are distributed. So these are laws that you're going to have to know. So we're going to watch this video today, and then we're going to come back and practice. You're going to get a practice sheet at looking at these. Okay. All righty. So back over here with Professor Dave. Sometimes Professor Dave talks a little fast, so let me see if I can slow him down a little bit. Nope, I cannot. Oh, yes, I can. All right, we're going to slow him down a little bit because he talks very fast. Okay.
Mendelian genetics.
He knows a lot about the science stuff. Professor Dave explains.
That's too slow.
Through a study of chemistry, biochemistry, and biology, we can come to know a lot about the molecular and cellular processes that generate and sustain human life. But long before we even knew that cells and molecules existed, we were able to understand the concept of heredity. Children look like their parents. Traits are passed on from generation to generation, be it hair color, skin tone, height, or anything else. For most of human history, we had no idea how this happened. And even with the biology we have just learned, it still isn't entirely clear. But a discussion of how traits are passed along from one organism to the next is a discussion of genetics. And to do any justice to this field, we must go back to its inception with a man named Gregor Mendel.
The data, there's too much of it.
That's okay. SAS-Viya
These
builds models and makes decisions, so you don't have to freak out.
But we like freaking out.
We're getting so much done!
From data to ta-da, SAS-Viya.
And that one.
We all have an origin
Some of them
story.
won't let me delete them.
One unique to us. Just like.
There we go.
Mendel grew up poor in an agricultural area of Europe and entered an Augustinian monastery at age 21. After some time there, he left to study science in Vienna and then returned to the monastery, eager to begin his research. In Mendel's time, it was thought that heredity had to do with a blending of attributes, like the way two colors mix to give an intermediate color. But this did not account for the way that traits can skip a generation. Thus, it seemed to Mendel that heredity must be based on discrete units that can be inherited, which came to be referred to as genes. In the gene theory, each parent has a set of genes, and these are retained intact
Okay, so
in
that's
the
one
offspring.
thing that he had. He had the gene theory of inheritance, that you inherit. It simply means that you inherit your traits from your parents. So that's a Mendel thought.
Mendel worked for several decades with pea plants in the garden of his monastery to produce an impressive body of data to support the gene theory, all of which was done decades before chromosomes were observed and understood. We will discuss the methods Mendel used to breed the pea plants and the conclusions he was able to derive from them. First of all, why peas? Well, the pea plant has a number of visible traits that can vary, so results are easy to observe. These are things like flower color, seed color and shape, pod color and shape, flower position, and stem length. Generations of the pea plant are short and the offspring are many, so data is easy to gather. And Mendel could easily control mating by removing the stamens, the male fertilizing organs that produce pollen, from a particular set of plants. Then he could deliberately cross-fertilize with pollen from other plants. This means he had complete control over which plants were mating with which other plants. Some plants were true breeding with respect to a particular trait, meaning that some plants with purple flowers, when self-fertilizing, gave generation after generation of entirely purple flowering plants. The same goes for the plants with white flowers. So this meant he could breed true breeding white flowering plants with true breeding purple flowering plants and see what happens. This kind of process is called hybridization, and we can call the two true breeding plants the parental generation, while the offspring will be referred to as the F1 generation, or the first field.
Okay, now we are going to be drawing trees like that, so it's important that you remember what he's talking about. So you're going to take pure plants and you're going to hybrid them. That means you're going to cross them to create something that is not pure. And the offspring is also always called the F1 generation, and F1 stands for first filial. Okay, like I said, there's a lot of vocabulary with this, and so I'm going to need you to be paying attention and not playing your little video games in the background like some of y'all are doing.
Filial generation, from the Latin word for son. If these F1 plants are allowed to reproduce further, we get the F2, or second filial generation. Mendel did experiment after experiment with different kinds of pea plants, following the traits expressed, or phenotypes, up to the F2 generation. From this mountain of data, he developed two fundamental laws of genetics. These are the law of segregation and the law of independent assortment. To see how these laws were derived, let's examine one of his experiments.
Dreamers, darers, story sharers,
Let's say purple flowering and white flowering plants are hybridized. We would find that the F1 generation is entirely purple. No white, no pink, just all purple. This experiment alone proves that any kind of blending hypothesis must be false. But even more perplexing is that if the F1 generation is allowed to self-pollinate, the F2 generation gives plants that are both purple and white in roughly a 3-to-1 ratio. This strongly supports what we now refer to as the gene theory, because it means that somewhere in the F1 plants there must have been some kind of information pertaining to white flowers that was not lost and was expressed later with the F2 plants. This white flowering gene must simply have been hidden or silenced in the F1 plants. We can therefore refer to purple flowers as the dominant trait and white flowers as the recessive trait. This combination of dominant and recessive traits was observed for many other aspects of the plant, and precisely the same 3-to-1 ratio was observed when examining the phenotypes in the F2 generation for smooth versus wrinkled seeds, yellow versus green seeds, green versus yellow pods, and so forth. The reason for this is that each trait corresponds to a particular gene, like a gene for flower color. But each gene has 2 versions, or alleles, in the genome, because the gene is found on 2 homologous chromosomes, one from each parent. These 2 alleles can be of a different genotype, meaning that the sequence of nucleotides are different. And this can lead to different phenotypes when expressed, like the 2 different colors that are visible on the flower.
All right, I'm going to slow this down. All right, so he's talking phenotypes. Again, vocabulary. Phenotype means what does it look like to the eye, physical appearance, okay? And then down here to the bottom, we're looking at what genes are present. So you see, you have the, remember when we did genetic codes and we had the sequence of nucleotides and we learned A and G and C and T and all that sort of stuff, okay? So we're coding for color or flower color, but we can code for purple or we can code for white, and they will have a different sequence. What he's saying here too is that the code for purple will always overshadow the code for white. So we call them dominant for purple and recessive for white. Okay, homologous chromosomes. Remember, they're the chromosomes that have the same information on them, even though, I mean, when I say the same, they're coding for the same characteristic, but they may code for it differently. But they're still homologous chromosomes. One from mom, maternal, one from dad, paternal. Okay, so like I said, there's a lot of vocabulary on this page alone.
Although Mendel didn't know anything about DNA, he was still able to decipher this concept of 2 alleles per trait. True breeding plants must have identical alleles for a particular trait. But when he performed the hybridization, he realized that one allele was dominant, in that it was expressed while the other was recessive and stayed dormant. In this case, when purple and white are hybridized, the F1 generation is all purple, so purple must be dominant. He even realized that each gamete must have only one of these alleles, such that when each plant is fertilized, one of any of the possible combinations of alleles will result. This is the law of segregation.
Oops. Hold on. I paused it too soon.
Dreamers, darers, story sharers.
We
Okay,
now
so
understand this
stopping
is true.
again. All right. So what he's saying, every organism has 2 alleles, and and then during meiosis, remember, one goes from mom, one of the 2 will go to mom, and the other one will come from dad. So we're going to be looking at the possibility of which one of the 2 you give away. In the case of the dominance, both, it doesn't matter which one they give, both of them are the same. Both of these are purple. On the case of recessive, both of these are white. So this flower can only give a recessive gene. This one can only give a dominant gene. But what happens when we fertilize is that the purple and the white come together, and the white doesn't disappear, but you can only see phenotype the purple, but the white allele is still there. Okay, so we're going to come up with a new vocabulary word for that. So the offspring have 2 alleles, but one is dominant and one is recessive. One is purple, one is white, and they all look purple because we can only see the dominant allele.
True, because we have learned about meiosis and the haploid daughter cells that result. In this way, Mendel was able to explain the phenotypes in the F2 generation by rationalizing that true breeding purples have 2 purple alleles and true breeding whites have 2 white alleles. So when the sperm from one fertilizes the egg from the other, all of the resulting plants in the F1 generation must have one of each allele. Because purple is dominant, they are all purple. But when the F1 generation produces its own gametes, some will be purple and some will be white. So when they self-fertilize, there are 4 possible combinations, and those are purple and purple, purple and white, white and purple, or white and white. Since purple is present in 3 of those, that explains the 3-to-1 ratio, given that only a plant with 2 recessive white alleles will appear white. This kind of logic can be displayed using Punnett squares. For these, we make a grid, and along the top boxes, we place the alleles for one plant. If we are allowing the F1 generation to reproduce, all of those have one purple allele, which we can symbolize with a capital P, since we capitalize the dominant allele, and the white allele will be a lowercase p, as it is recessive. The other plant will have its alleles to the left of the boxes. Then we just populate the boxes to form all the possible pairing combinations. Dominant alleles are listed first, and there is the distribution just as we described before.
Okay, now what they mean by populating the boxes. Let me come back over here. All right, you're going to take one letter from each side and put it in the box. Then you're going to take this letter and that one up top and put it in the box. And same thing over here, you're going to put those up, this top one with the bottom one and this top one with this bottom one. So that's how he populated the box. Okay, and you got to be careful, and then you all need to do this too, because when you do worksheets for me, it's going to be real important that you are particular about making capital letters and lowercase letters, because it matters the way it shows up in the box. Okay.
Alleles are listed first, and there is the distribution just as we described before, with a 3-to-1 ratio of purple to white in the F2 generation. When an organism has identical alleles for a particular gene, it is said to be homozygous for that gene. If it has one of each allele, we call it heterozygous. As we can see, homozygous dominant and heterozygous both result in the dominant phenotype. Only homozygous recessive results in the recessive phenotype being visible in the plant. For this reason, we must understand that observing the phenotype that is visible for a particular trait in an organism does not tell us with certainty what genotype is present, as multiple genotypes result in the same phenotype.
Dreamers,
Too many commercials.
darers, story sharers, multitask.
However, we can breed this organism with another organism that has a known genotype for that trait and analyze the phenotypic distribution in the resulting generation to decipher what the unknown genotype must be. When Mendel discovered the law of segregation, he was looking at one trait at a time, which we would call a monohybrid cross. But when he started looking at 2 traits at the same time, he discovered the law of independent assortment. He knew that the seeds of the pea plant could be either yellow or green, with yellow being dominant, and they could either be round or wrinkled, with round being dominant. He took true breeding plants with round yellow seeds and crossed them with true breeding plants with wrinkled green seeds, performing a dihybrid cross. As we would expect, all the plants in the F1 generation end up heterozygous for both traits, thus exhibiting the dominant phenotype for both traits. But because we are examining 2 traits at once, there are now 4 possible combinations of alleles produced in the gametes of the F1 plants. If these plants reproduce, we end up with a variety of possible genotypes for the F2 plants, because the Punnett square must involve all possible combinations of all possible gametes from 2 different plants.
And you will have to learn how to do this. We will definitely start with monohybrid crosses, because those are easier. And once you wrap your head around a monohybrid cross, then we will move to a dihybrid cross. And the thinking behind this is that on a chromosome, they don't just have one gene. There are several genes. So like you said, the gene for flower color, the seed texture, the seed color are all on a chromosome, and how they distribute themselves is what he calls independent assortment. They don't all travel together. And during meiosis, when we have that separation of chromosomes, one might go here and this one might be here and this one might be over here. Since there are 4 different cells that are produced at the end of meiosis, each one of those 4 could have independently one characteristic over another one. So we could have 4 different cells with 4 different characteristics in them.
The phenotypic distribution for a dihybrid cross will always be 9:3 to 3:1, where 9 represents the proportion of the F2 generation that will exhibit the dominant phenotype for both traits. 3 represents the proportion that will show dominance for one and recessive for the other. The other 3 is for the reverse situation, and the 1 is for the ones that will be recessive for both. These results are significant because they show that each trait is determined individually. Just because the F1 generation was heterozygous, it doesn't mean that the F2 will be. The alleles for each trait separate at random during gamete formation, thus demonstrating the law of independent assortment, and also demonstrating that the combination of alleles that occur in any given organism is determined entirely by probability. 16 possible combinations, 1 in 16 chance for each one, so roughly 1 in every 16 plants will exhibit a particular genotype.
Like President Trump, I don't owe anybody anything. Georgia can lead America in criminal
All right, let me see if I can. That is so wrong that they put that on
deportations.
There. Okay, let me exit out of that. Is that they're not supposed
Cut
to do,
income taxes
they're
in
not
half.
supposed to put political things inside your videos like that. All right, so I'm going to stop it there anyway. So while I do attendance, I want you to go back and look at, let's see if there's a way I can clip that out of there, because that is just wrong. But anyway, all right, so read over this page again. They talked about all these laws down here, so we're going to practice those. And what time is it? Okay, we have a few minutes. All right, so go to the next page, because I'm going to give you a sheet so that you can do an initial practice of what a monocross is, and we'll do dihybrid crosses next time. But look at this one while I do attendance, because it's getting late. I need to stop and do attendance. Okay. All right, and then do read them, because like I said, this is not an easy topic to do, and completing these worksheets gets a little complicated. So if you're not paying attention, you don't pay attention to, because these are word problems. All of these worksheets are going to end up being word problems, where they will give you a scenario, they will tell you what the characteristics are of the parents, and you have to draw it out in the Punnett square. So if you're not familiar with the vocabulary, you will not draw your box correctly. So please do read that page. And it's not going. Okay. Okay, you were reading the page. That's what you were doing. But I'm about to, hopefully you were reading the page. And now I'm going to show you what you need to do.
I was reading all the pages.
Okay, I'm good. I'm ready. Alrighty. Hopefully you weren't the only one. Okay. So we're going to do, so I got some more reading for you to do. All right. There is a worksheet down here to the bottom. When you open up that worksheet, it's going to tell you to get your information from the links above. And rather than, okay, let me show it to you this way because it's still loading on your side. All right, so the worksheet's going to look like this once it loads. Okay, up here at the bottom. So I don't want you to click on the tutorials. I've printed those out for you in the box. So based on reading what's in the box, which still hasn't loaded. All right, so if you look at the assignment, I'm going to put this up as a student view so you can see it as you and not me. All right, so you're going to read first. So these are the two sets of information that you are going to base your information on. So you don't have to go clicking anywhere and run into a website that is blocked. And this is the worksheet down here. All right, so hopefully it's loaded now. Nope, not yet. All right, well, while that's loading, I'll just switch over here. All right, so again, based on the reading that I already have in the box for you, you're going to fill in the blanks. So from those passages, you're going to finish these 3 sentences. Okay, and then you're going to also, based on those readings, you're going to circle the correct answer for these 4 questions. And then after you read this passage, these are the lovely
Where's it
little
under?
boxes. Say what?
What is it under?
Right now it should be on your home page. So let me back up and, like I said, it probably hasn't loaded yet. So right now, just follow me. Just follow me. It's because the page is still loading, apparently. All right, so just like in the video, you're going to practice taking these letters and putting them in the boxes, okay? All right, so that's what you're going to, so for these, they have put, for the first 3, they put the letters of the parents on the outside. For the next set, they want you to, based on who you have in here, they want you to guess who the parents are going to be. So I'll give you a little hint. If you got a capital R here, then you had to have a capital R over there. If you had a capital R, and if 2 capital R's in this box here, you had to have 1 up there. So whatever's in the box had to come from the boxes on the side. So that's how you're going to guess at what the parents are going to be. So again, the first 3, they've telling you who the parents are. They want you to tell who the offspring will be. Over from 4 to 8, they have not told you who the parents are, but they've told you the possible, actually number 7 tells you who the parents are. So you just have to figure out the offspring, but number 8 also tells you just who the possible offspring are, and you have to figure out who the parents are as well as figure out what the other ones will be. All right, so let me come back over here, and it's still loading, but give me a minute, or give it a minute. But this should be on your, so yeah, that's, where is it? It is, if you refresh your page, it is right here, Mendelian genetics. So that'll take you to that. And I'm going to add it to the module as well. All right, so it's on your home page if you refresh. And then I'm also going to add it in in the module. Okay, so we are in genetics. And we were looking at, I'll put it after this one. Oops, that's not what I want to do. Okay, I want to add it to the module. Okay, so now it's in the module right here. So if I click on that in the module, it's going to take me to the assignment. So again, you're going to read this. You're not going to click on the tutorial like it said. Let's see if it's loaded yet. Yay, it's loaded. Okay, so those are hyperlinks, but more than likely they'll block you. So don't try to. What's on these two links is what I put on the first page. So you're going to read what's in the text box for the assignment and then answer the questions. Again, same thing with those, ignore those. So based on what you are reading here, you're going to answer the questions on the worksheet. Okay. All right, so you can kind of download that and kind of wrap your head around it. At least do the reading right now. You don't have time to do anything else. And we will continue working on this next class. So again, download the worksheet and then come back and start reading here, because the answers that go in the boxes or on the first page come from the reading this page here. Okay, I'm going to say that again. All right, when you download the worksheet, that's the first thing you need to do. You have, it says based on the tutorial, ignore those hyperlinks. I have already reproduced this information for you on the first page. So this is kind of like a fill in the blank. The first three are like fill in the blank. So you're going to get the answer from reading the first page. And these are multiple choice, but you're still getting the answer from reading the first page. Same thing here. Ignore the hyperlink. You're going to complete, put this in order based on what you have read. So you're going to put numbers here, 1 through 5. They've already told you that this is the 5th one. So you're putting 1 through 4 in the spaces here. Okay, and then you're going to complete the boxes the way we were watching it in the video. All right, so at least get through the top part. This part down here, we may have to do together, but do as much as you can. And then we'll see how you do on these boxes. But we're going to be practicing these boxes a lot. All right, so I'm going to stop talking so you can start reading. And all of these vocabulary words that are in blue, you have those on your vocabulary page at the beginning of Canvas, so you don't have to click on those hyperlinks either, because they're just definitions. You have all of those definitions on your introductory page too. Let me go back and share that as well, so you know what I'm talking about. All right, so all of these little blue things here, those are vocabulary. If you go back to your overview page when you started, all of those vocabulary words are there. So you do not have to do any of those hyperlinks. They are already
Miss Harris.
in.
For the first.
For the first what? Change your mind on the question? Or do I need to go to the...
Rewrite the whole thing on there?
You mean in this space you need to finish the sentence? Yes. All right, let me... let me go back to there. Let me go back to home page. I find the fastest way to get to these. Why is it not on my home page? It was as a student. There we go. Okay. Let me repeat that. All right, so that's the first thing they said at the top. That's number one. Okay. No, you don't have to finish the sentence. All right, I'm telling you that's number one. Then produced offspring right around seeds. So you're gonna come back over here. That's the next one. And then and that's the third one. So basically it would be 1, 1, 3, 2. If you were putting them in order. Okay. You really didn't have to write anything. You just had to put them in order as to where they fell in your description here. Same thing with the other one. Then you have to answer these questions. This one also wants you to put it in order. Just put numbers. So over here, it's just said put the blocks of text in order, correct order. So that, again, I'm just telling you the answer. 1, 3, 2. If you were reading this in correct order, the way it is on the passage, you answer these. Okay. All right. Any other questions? Because it is time to go. Almost. Almost. Got about 5 minutes. Any other questions? Any other questions? Okay, so if you're in a good place and you have no questions, I think you know what you're doing. I think you know what you're doing.